Hello everyone and welcome to Let's Crack NEET UG in English. This is the channel where we are right now, and we will be starting with a new chapter in the unit of plant physiology. So let me see how many of you are there already. And before we get started, let us check for the audio. Excuse me. So, is the audio clear? Can you hear me clearly? Hello everyone, can you hear me clearly? So if the audio is clear, then we can get started. I can see some of you there. Just let me know about the audio first. I guess nothing is mentioned about the audio. That means everything is clear. And before we move ahead with this chapter, let me just go over the basic things. The first thing that we will be talking about is the types of respiration, aerobic, anaerobic, okay? Like we are on this channel, that is Let's Crack NEET UG English. And for the new students, this is some information about me. I'm your biology teacher, Dr. Neela Bakore, and we are using India's largest learning platform, that is Unacademy. And if you have not installed this app yet, please go ahead and install this Unacademy learning app, because on the app there are many features which would be available to you, and you will be able to take advantage of those things. Like there are live classes which are conducted by various teachers. You will have unlimited access to all those like live lectures. Plus there are quizzes which are conducted. Okay? The live classes are conducted by various educators from various fields, like there are physics experts, chemistry experts, and then you can study from them. These are few names of the top educators, and this is the change which has taken place already. Today is 24th, and I told you that from October 24th, this subscription plan hike is going to take place, and now you will be required to pay this much amount. But you can still get 10% discount if you use this code, that is Neela. Now we are talking of types of respiration. So before we start, again, I hope the audio is clear and I'm audible clearly to everyone. If there is any issue, just let me know. Otherwise, we can get started. Alright, so nobody is writing anything, that means the audio is clear. Now when we talk of types of respiration, what exactly we mean here by respiration is the cellular respiration. Now when we use the word respiration, there are two parts. One is the breathing part, which is like the external respiration. So when we breathe in, there is gaseous exchange, then the oxygen is transported by blood, and now oxygen has reached up to the cell. So at the cell level, using that oxygen, some organic matter like glucose is being broken down to release ATP. And this is what is meant by cellular respiration. So when we talk of this respiration, then there are two types. One is called aerobic respiration, and as we all know, aerobic respiration takes place in presence of molecular oxygen. Then if such type of respiration is what we are talking about, that means breaking down of glucose in presence of oxygen, then that is called aerobic respiration. And if breakdown of organic matter like glucose is taking place in absence of oxygen, then that will be called anaerobic respiration. That is, this is the process which takes place in absence of oxygen. Then we call it anaerobic. So these are the two main types of respirations, aerobic and anaerobic. We will be talking about anaerobic respiration, the different types of anaerobic respiration in today's class. But what all happens in aerobic respiration? This is just to give you a little idea or heads up of aerobic respiration in presence of oxygen. So first step is glycolysis. Then there is an intermediate step or link reaction which is known as oxidative decarboxylation. It is a link reaction basically between glycolysis and Krebs cycle. So the next is Krebs cycle, and fourth is electron transport chain, and the fifth is oxidative phosphorylation. We have talked about this word phosphorylation. Phosphorylation is addition of phosphate, but when we were talking of photosynthesis, we talked of photophosphorylation, because phosphorylation, that is addition of phosphate, was taking place in the presence of sunlight or due to sunlight. But here this phosphorylation is taking place normally without that sunlight. Okay, so there are students who have joined in little late, and just those who joined late, we are starting a new chapter in plant physiology, and that is respiration in plants, and we just started with types of respiration. And today, before I end the class, I have to give you the timing for this class, because for this week our class is going to be slightly at different time, and I'll tell you the dates and everything. Hiren, good evening Hiren, how are you? Is the audio clear? This is just the beginning. We just started the class with the types of respiration, just giving you a basic idea. Okay, so the audio is clear. We have started a new chapter in plant physiology, that is respiration in plants, and when here we are talking of respiration, so we mean cellular respiration, that is breakdown of glucose inside a cell. Now if it takes place in presence of oxygen, we call it aerobic, and these are the 5 steps. This we will take up starting from the next class, and today we will be talking about only anaerobic respiration, that is in absence of molecular oxygen. So now when we talk of anaerobic respiration, there are 2 types of anaerobic respiration. Or if I have to just give you something more about anaerobic respiration here, we can write that anaerobic respiration is known as fermentation. Now normally when people talk about anaerobic or rather fermentation, they are talking only about alcohol formation, but in general anaerobic respiration is known as fermentation, and we can classify this fermentation into 2 types, homo fermentation and heterofermentation. Homofermentation and heterofermentation. Now what is meant by this? Homofermentation is when only one product is obtained. Only one product is obtained. But if you are talking of heterofermentation, then more than one product are obtained. So as we talk of lactic acid fermentation and alcohol fermentation, we will also realize whether it is a homofermentation or heterofermentation. Okay, Hiren is saying this timing is good, doesn't clash with our school timings. So have your schools begun? Have you started going to the school, Hiren? Because at our place here, I am based at Indore in Madhya Pradesh. Here the schools have not opened yet. Oh, your schools are on. So are you attending like the regular classes on a daily basis? Oh, online. Okay. All right, got it. Online classes. All right, so your online classes are taking place and you are free by this time. All right, now I got it. Because here also only online classes are going on and the students go to the school only if they have some kind of a difficulty which they need to discuss with their teachers. All right, so I guess everywhere in India it is pretty much the same. Okay, so we will come back to this homofermentation and heterofermentation. So now let us talk of the first type of anaerobic respiration. It is known as lactic acid fermentation. What happens in lactic acid fermentation is, first, glucose is broken down into two molecules of pyruvic acid. Glucose is a 6-carbon compound and pyruvic acid is a 3-carbon compound. So there are two molecules of pyruvic acid formed. This process is known as glycolysis. That means glycolysis is a step which is common for aerobic as well as anaerobic respiration. Glycolysis is the first and common step for aerobic and anaerobic respiration. And next, or the second important thing is, in this step there is no requirement of oxygen. That means glycolysis is an anaerobic step. So in this particular step there is no oxygen required. So now there are two molecules of pyruvic acid formed. This pyruvic acid gets converted into lactic acid. Lactic acid is also a 3-carbon compound and two molecules of this are formed. So in this reaction there is only one product which is formed. That means this is homofermentation. Alright, so if in a reaction only one product is formed, then you will call it homofermentation. Two lactic acid molecules are formed. And in this particular thing, what happens is, if you have studied little bit of glycolysis or aerobic and anaerobic, you will understand this. We can write this in a different manner also. Glucose, two molecules of pyruvic acid, same reaction I am writing in a different manner. then these 2 molecules of pyruvic acid change into 2 molecules of lactic acid. Only way of representation is different. This step is glycolysis. Now in glycolysis, if you have studied that, in glycolysis NAD gets converted into NADH2. It is not NADP. NADP is used in photosynthesis. When pyruvic acid gets converted into lactic acid, this NADH2 gets oxidized and pyruvic acid is getting reduced into lactic acid. So here the enzyme is, you can call it reductase or dehydrogenase. And for this enzyme to work, there is a coenzyme required. So here it is dehydrogenase, and every enzyme, or rather most of the enzymes, they require coenzymes. Coenzymes are substances which are going to help them help in the reaction. So that is FMN, flavin mononucleotide, and an activator is also required, and that is zinc ion. So for this reaction to take place, an enzyme is required. That enzyme can be called reductase or it can be called dehydrogenase. FMN is the coenzyme and zinc is the activator of this enzyme. Okay, so this is one type of anaerobic respiration. Now where does this take place? This type of respiration, that is lactic acid fermentation, takes place in our muscle cells during excessive exercise or rigorous exercise. During that time, because as soon as we start exercising, the initial breakdown is aerobic because oxygen supply is there. But if we are continuously working, then that oxygen supply may not be enough. So glucose is broken down in absence of oxygen. And in this process, in this particular step, we will get 2 ATPs. So in anaerobic respiration, like lactic acid fermentation, we are going to get only 2 ATPs. So 2 ATPs are obtained, only one product, and this is one place where this is taking place. The second is RBC. Our RBCs are enucleated and without organelle. First step, that is glycolysis, takes place in cytoplasm. But other than that, mitochondria is required. But because in RBC there is no mitochondria, there is no aerobic respiration beyond this. And that is why in RBC, anaerobic respiration takes place. So this is about lactic acid fermentation. So can we say that NADH2 is kind of trapped in the cycle and cannot be used anywhere else? Yes, we can say that, but this NADH2 which is formed, this one, is actually worth 3 ATPs. If the complete process, like in aerobic respiration, if this NADH2 reaches up to the last step, then it is going to give us 3 ATPs. When we come to glycolysis in detail, we will say that we are going to get 2 NADH2 molecules. Here there are 2 NAD, 2 NADH2, both are used up. So basically it is like a check. You are getting a check. 1 NADH2 is a check of or for 3 ATPs. But for that you will have to put that check in the bank. Then only it gets encashed. And here what is happening? That check is given, released, and it is taken back. So those ATPs which the check is worth, those ATPs are not obtained. Is this part clear? Is this lactic acid fermentation part clear? So we will take the next type of anaerobic respiration. Okay, so this is clear. Let us go to the next type, that is called alcohol fermentation. Now in alcohol fermentation, the first part remains the same. That first part is that glucose, which is a 6-carbon compound, is broken down into 2 molecules of pyruvic acid. Pyruvic acid is a 3-carbon compound, so we have 6 carbons. This step is glycolysis, and in this step 2 NAD have given us 2 NADH2. So this is common, whether we are talking of lactic acid fermentation or we are talking of alcohol fermentation. Now in the next step, pyruvic acid, which is a 3-carbon compound, breaks into 1 2-carbon compound and 1 single carbon. So what is that single carbon? It is lost in the form of carbon dioxide. 2 molecules of carbon dioxide are released. 2 carbons are gone. Total left are 4. So we will get 2 molecules of acetaldehyde. This is a 2-carbon compound. So 2 carbon, 2 molecule, 4. These 2 carbons lost in the form of carbon dioxide. The enzyme which is here is called pyruvate decarboxylase. Decarboxylation is taking place, and for this reaction to take place, this enzyme requires a coenzyme. It is called thiamine pyrophosphate. Thiamine pyrophosphate. And zinc ions are required. These are the activators. So this is a coenzyme of decarboxylase, and this acts as activator. Alright, so 2-carbon compound is formed and carbon dioxide is given out. Now this acetaldehyde changes into ethyl alcohol. Ethyl alcohol is 2-carbon compound, C2H5OH, 2-carbon compound, and 2 molecules of this are formed. So 2-carbon compound, 2 molecules means 4 carbon, but for this reaction to take place, this NADH2 gets used up. So again, here in glycolysis, there are 2 ATPs given out. So when we talk of alcohol fermentation, what are we getting? 2 ATPs. What did we get in lactic acid fermentation? 2 ATPs. What was the product in lactic acid fermentation? Only lactic acid. What are the products here? One is ethyl alcohol and second is carbon dioxide. So this is heterofermentation. So there can be homofermentation or heterofermentation. Alright, so depending upon how many products are formed, single or more than one, and in both the cases the amount of ATP or the number of ATP molecules obtained is only 2. So in anaerobic respiration, ATP obtained are only 2. Only 2 ATPs are obtained, and that is why this process is less efficient. You are breaking one glucose molecule, but you are getting only 2 ATPs. So that is why it is considered as less efficient method. Okay, activator and coenzyme. Enzymes need some protein molecules which are going to help them, some non-protein molecules which are going to help them, and some mineral ions which are going to help them. So the coenzymes are organic molecules which are temporarily attached to the enzyme. This we will discuss when we come to the biomolecule part, and we will be talking about these enzymes in detail. So if some organic substance is attached to the enzyme temporarily, we call it coenzyme. If it is permanently attached to the enzyme, it is known as prosthetic group. If it is just a mineral ion which gets attached and detaches, that is called an activator. Alright? So are these two types of anaerobic respiration clear? That is lactic acid fermentation and alcohol fermentation. And where is alcohol fermentation takes place? It takes place in yeast. And that is why whenever there is yeast added, this type of reaction takes place. When alcoholic beverages are to be made, that time same yeast is used, because yeast is going to use the carbohydrate present in whatever substance we are using and will convert it into ethyl alcohol. Alright, so it is clear, that's very good. Now let us come to the next thing. This is called respiratory quotient. I'm sure you've heard of IQ, intelligence quotient. Now what exactly we mean by intelligence quotient or IQ? Some like if there are a couple of students, somebody would say this person has more IQ and this person has less IQ. In the class also sometimes it happens that you feel that this person or this student has greater IQ and somebody else has lower IQ. What exactly is this IQ? See, the information is going into your brain. Now, people who are able to interpret, analyze that information in a better manner, if they are able to apply logic and interpret it in a better manner, then they are able to produce better results. And we say that IQ is good. So it is basically what is the input and what is the output. Information flows in, this is input, and the exam and the result, that is the output. So what happens inside your head is actually the interpretation or the analysis part. Here we are talking about respiratory quotient. How do we define it? It is the ratio of volume of carbon dioxide evolved, that means given out, to the volume of oxygen consumed during respiration. So how much oxygen is consumed? This is input. How much carbon dioxide is given out? This is output. And we are talking in terms of respiration. So we are calling it respiration quotient or RQ. And RQ depends on the substrate. What is that substrate which is being broken down? Like if it is glucose which is broken down, RQ is going to be different. If protein is going to be broken down, RQ is going to be different. So what is the substrate which is being broken down to release energy? That is what is respiration. Now, how much energy is released? That will depend on the substrate, or in other words we can say that RQ depends on the types of substances or substrate. Alright, so if we have to calculate RQ, we can write a very simple formula, that is volume of carbon dioxide evolved upon volume of oxygen consumed or used up. So again, whenever there is quotient, it is a comparison. How much is the inflow and how much is the outflow? And that will decide the quotient. If it is on the basis of our intellect, that is our knowledge and interpretation, then we call it IQ. Here, because we are talking of respiration, we are calling it RQ. So how do we calculate in the RQ? For that, we need to know a reaction. Okay, so if you know the reaction, you have to just place the values in this particular formula and you will get the RQ. So what I have done is, I have written down the reactions for you which are balanced. So the first thing first is we are going to calculate the RQ of carbohydrate. Carbohydrate means glucose. So C6H12O6 plus O2. This is aerobic breakdown, and carbon dioxide, water are given out. Plus energy is also produced. Now if you balance this, it is going to be 6O2, 6CO2 and 6H2O. Our formula is RQ, volume of carbon dioxide evolved upon volume of oxygen used up or consumed. So here, how many carbon dioxide molecules are there? We write 6, evolved. And how many molecules of oxygen were used up? 6. So here it is 1. Now the most important thing or the statement which we have to remember is RQ of carbohydrates is 1. This is the statement which we have to remember. This is the most important thing. Is it clear how did we calculate the RQ of carbohydrate? This is glucose, which is a carbohydrate. So you need to know the reaction, that if that particular carbohydrate is broken down to release energy, that is in respiration, then how many molecules of oxygen are required and how many carbon dioxide molecules are given out. So if this is clear, then I'll give you one more equation. Is this part clear? This is RQ of carbohydrate, and this is standard. All other questions which will be asked to us, we will be answering using this statement. So whenever there is a carbohydrate, RQ is going to be 1. So if now I ask you that if you're eating potatoes, what is the RQ of that food? What is there in potatoes? Potatoes have starch, and starch is carbohydrate. That means RQ is going to be 1. Simple. So the question which can be asked is, what is the RQ of carbohydrate? Straight question. Or the question can be twisted by giving you the substance that you're eating or the food product which you're eating. So this way, two concepts are evaluated. One, do you know whether potato has starch and starch is carbohydrate? This is one part. And two, do you know that carbohydrates have RQ as 1? So there are two concepts which are checked in this particular type of question. Now let us come to the next one, and I'm going to write this balanced equation for you. It is I'm writing the equation for a fatty substance. It is 2 molecules of palmitic acid, C51H98O6. This is tripalmitin or palmitic acid. It reacts with 145 oxygen molecules and you get 102 carbon dioxide molecules, 98 water molecules and energy. Okay, our formula for RQ is carbon dioxide evolved upon oxygen consumed. So it is going to be 102 upon 145. What is going to be your answer? Is it going to be 1? Is it going to be more than 1? Is it going to be less than 1? See here, It is not asked that it is 0.7, 0.8, 0.9 or 1.5, 1.6. They just want to know whether it is 1, more than 1, less than 1. What is the answer? Okay, so it is going to be less than 1. Here it is going to come very close to 0.7. Okay, so now our statement, RQ of fats and proteins is less than 1. This is our second statement which we have to remember. So RQ of carbohydrates was 1, always, whichever carbohydrate you talk of. RQ of fats and protein is always going to be less than 1, depending upon the fat, depending upon the protein. It can be 0.7, 0.4, 0.5, anything, but it is always going to be less than 1. Alright, so this is the second one. Now let us talk about one more, RQ of organic acids. Again, I am giving you one more equation, and I have balanced it for you. 2 molecules of oxalic acid undergo aerobic breakdown to give you 4 carbon dioxide molecules, 2 water molecules and energy. Okay, our formula for RQ is CO2 upon O2. So here it is 4 upon 1 molecule. So 4 molecules of carbon dioxide and 1 molecule of oxygen. So how much is going to be the RQ? More than 1, less than 1 or 1? It is going to be more than 1, is more than 1. So this is our third statement. First statement was RQ of carbohydrate is 1. RQ of proteins and fats is less than 1. And RQ of organic acids is more than 1. Alright, so here we are not really worried about whether it is 0.6, 0.4 or what. Here we need to know whether it is 1, more than 1 or less than 1. Let us talk about one more reaction. RQ under anaerobic condition. Just now we wrote an equation where glucose undergoes anaerobic breakdown. So if glucose is undergoing anaerobic breakdown, then that means there is no oxygen required. So if we have to write an equation, then it is, say glucose is what we are writing, C6. I need a different darker color. Okay, it is C. Let me choose the color, a darker color. Okay, so it is C6H12O6. This is glucose and it is being broken down in absence of oxygen. And what do you get? You get 2CO2 plus 2C2H5OH plus 2 ATPs. This is what we got in anaerobic respiration. So now how do we calculate RQ? CO2 upon O2. How many carbon dioxide molecules? 2. And how many oxygen? 0. So what is going to be the answer? You divide any number with 0, it is infinity. So now we have one more sentence, that RQ under anaerobic condition anaerobic conditions is infinity. And now we know how we have calculated this, okay? So this is our next statement. So far, carbohydrates give us 1. Proteins and fats give us less than 1. Organic acids give us more than 1. And under anaerobic condition, it is infinity. Now let us do one more. In succulent plants are the plants which grow in desert areas like cactus. So in cactus, there is a different kind of reaction which takes place. So I'm going to write that reaction for you. 2 molecules of glucose, that is 2C6H12O6 plus 3O2. In presence of oxygen, it will be broken down into malic acid plus water plus energy. Our equation or formula to calculate is carbon dioxide upon O2. So here carbon dioxide is not given out, so that is 0. And we are using 3 molecules of oxygen for this reaction. So what will be the RQ in case of this? This is going to be 0. So this is our 5th statement, that RQ in succulent plants is 0. We have to remember only these sentences. And if we remember these sentences, then we will be able to solve the questions. So far, is there anything which is difficult, anything which you have not understood in this particular part? We will go over all those things which we have talked of so far. Okay, we started with types of respiration. This is cellular respiration, and this is our 2nd chapter in the unit of plant physiology. 2 types of respiration, aerobic and anaerobic. Aerobic is in presence of oxygen, anaerobic is in absence of oxygen. Anaerobic respiration is normally termed as fermentation. And then there are 2 types, homofermentation or hetero, depending upon how many products are obtained. Aerobic respiration is a complicated process, and there are 5 steps. First is glycolysis, then there is a link reaction which is called decarboxylation. Then Krebs cycle, then there is electron transport chain, and then oxidative phosphorylation. So this part we will take up later on. But right now we are talking of anaerobic respiration. Lactic acid fermentation. Lactic acid is a 3-carbon compound. So glucose is first broken down into 2 molecules of pyruvic acid. Pyruvic acids are 3-carbon compound. And these 3-carbon compounds change into lactic acid, which are also 3-carbon compound. So 6 carbons from glucose into 2 molecules of 3-carbon compound, that is pyruvic acid, gets converted into 2 molecules of 3-carbon compound, that is lactic acid. So here there is no carbon dioxide given out, only lactic acid, only one product is produced. That means this is homofermentation. Okay. Where does this take place? It takes place in our muscles when we are doing rigorous activity or exercise. Just by walking or just doing simple work, this will not start. And when do you realize, suppose you're jogging or you're running, after some time you feel that your muscles are getting like stiff and they're not working, which is known as muscle fatigue. And muscle fatigue is caused due to accumulation of lactic acid. So this one additional thing we will write here. Muscle fatigue is caused due to accumulation of lactic acid. And that is when you feel like that you should stop running, you wait for some time, and then all that lactic acid is taken away from that muscle. It goes into the liver where it is broken down further. And then the muscle starts working again. Okay, there is a question from Ayush. Ma'am, what is the difference between facultative and obligate aerobes and anaerobes? Yes, it's a very good question, and there are many students who find difficulties in this. I'll take that in a minute. Then alcohol fermentation. Alcohol fermentation takes place in yeast, and this is completed in one extra step. So 3 carbon containing pyruvic acid is first broken down into 2 carbon containing acetaldehyde. This is the step where carbon dioxide is released. So here there are 2 products. One is carbon dioxide and finally ethyl alcohol. So this is called heterofermentation. Then we came to RQ, respiratory quotient, that is amount of carbon dioxide evolved. It is a ratio to amount of oxygen used up. Simple formula, and it depends on the substrate. What is your substrate? If it is carbohydrate, the RQ is 1. And how do we know? We write the simple equation, and that is, we have to balance the equation. Then only we will get the RQ. For proteins, the RQ is always less than 1, be it protein, be it fat. For organic acid, RQ is always more than 1. In anaerobic condition, no oxygen is used up. So it is going to be infinity because there is some carbon dioxide evolved, no oxygen used up. So some number upon 0, it is going to give you infinity. And now in succulent plants. In succulent plant, there is a different reaction which takes place. So the RQ which we get is 0. Okay, now I'm coming to the question which is asked by Ayush, and I'm going to explain it here itself. Okay, let us talk about obligate or aerobes. What are aerobes? The organisms which require oxygen. Now these aerobes can be obligate or facultative. So we call it obligate aerobe and facultative aerobe. Obligate means, and we are talking of aerobe, that means we are talking of oxygen requirement. So if you use the word obligate, that means these microbes or these organisms, it can survive only in presence of oxygen. It can survive only in presence of oxygen. This means that if oxygen is not there, obligate aerobes will die. Is this part clear? Oxygen is a must. They will survive in presence of oxygen only. What is facultative? It can survive in presence of oxygen also. So obligate aerobe is the one which survives only in presence of oxygen, and facultative aerobe is it can survive in presence of oxygen also. That means if oxygen is there, then also they can survive. Otherwise, how do they survive? That means they are normally anaerobic. They are normally anaerobic. So they lead their life without oxygen. But if oxygen is there, they will still survive. So they can survive in presence of oxygen also. And obligate are they can survive only in presence of oxygen. Now if we have to talk about anaerobes, then can you tell me what will be obligate anaerobe and what will be facultative anaerobe? Kavya is saying respiration NCERT first portion not covered. Why? What is given in the first portion? What is in the first portion? Kavya, can you specify? What is in the first portion of NCERT in respiration? Okay, by the time Kavya writes the answer to that question, Hiren has written that obligate anaerobes can survive only. See, when you are using the word aerobe, you have to have the reference of oxygen. You cannot be talking about carbon dioxide. So obligate anaerobes are the ones, they survive in absence of oxygen only. That means if oxygen is present, they will die. So they survive only when there is no oxygen. And what will be facultative anaerobes? They can survive in absence of oxygen also. That means if oxygen is not there, still they can survive. So otherwise, how are they surviving? So these ones are normally aerobes. So normally they lead their life as aerobes. They use oxygen and survive. But they can survive in absence of oxygen also. So is the difference clear between aerobes and anaerobes? That was question from Ayush. Obligate anaerobes are those that can survive only in absence of oxygen. Very good. Kavya says NCERT, glycolysis and fermentation. When did you join the class, Kavya, today's lecture? What time did you join? Ayush, clear? Very good. Now, see Kavya, the class started early and after this lecture is done, you go through this complete video. Okay? So for you, I am taking you back to the first part. Because you started very late, that's why you don't know what exactly we were talking about. See, types of respiration, this is what we started with. Aerobic and anaerobic. Then we came to types of anaerobic respiration. And do you see glycolysis written here? Do you see fermentation written here? Glycolysis written, fermentation written, right? So we talked about all these things. No need to feel sorry, but I guess this is the class where the timing was a different one, and probably that's why you could not join in the beginning. But after this class is over, this video will be available on the chat on YouTube. So do watch the complete chapter, right? Your doubt of that initial glycolysis and those things will be cleared. And then we talked about this RQ. This is also very important if you have not understood that. So go over this. And now I'm giving you a sort of a practice thing. You need to find out what is the RQ of these things. Suppose you are taking a balanced diet. What all things are there in a balanced diet? Like I'll help you in one. What is there in balanced diet? In balanced diet, and balanced diet changes according to the age. So there is carbohydrate, there is protein, there is fat. So if you're taking balanced diet, what will be the RQ of that balanced diet? We know RQ of carbohydrate is 1. RQ of protein is less than 1, and RQ of fat is also less than 1. So what is the answer that we will get, or what will be the RQ of complete balanced diet? It is going to be, RQ here is going to be less than 1. Very good. Hiren has given the answer. Okay, because RQ is given at the end of it. But this is very important. That's why we take it in the beginning. Alright, can you tell me the answer for the second one? You had cheese sandwich. What will be the RQ of that substance, that cheese sandwich? 1, more than 1, or less than 1? RQ of cheese sandwich. What will be the RQ? 1, less than 1, more than 1. Ayush says less than 1. Why do you think, Ayush, it is less than 1? Because cheese is fat, and in sandwich there is bread, which is carbohydrate. So this is less than 1, and this is 1. So your answer is correct. That is, RQ is less than 1. Okay, you had rice and dal. Now what is the RQ of this? Less than 1, cheese sandwich. Correct. Less than 1. Okay, you had rice and dal for your lunch. What will be the RQ of that meal? RQ of rice and dal. Less than 1, rice and dal. What is rice? Carbohydrate. What is in dal? Is protein. So carbohydrate is 1, protein is less than 1. So again RQ is less than 1. Rice contains carb, dal contains proteins. Perfect. Potato chips. RQ of potato chips. Less than 1. Why do you think potato is starch? Potato is starch. That is carbohydrate. Why do you think it is less than 1? Ayush also saying less than 1, and Hiren also saying less than 1. Why do you think it is less than 1? Because potato is starch. Dr. Prem Chandran is also saying less than 1. Hiren now has changed to 1. Potato is starch. So why initially you thought that RQ is less than 1? Potato chips less than 1. My question is why? Why is it less than 1 when potato is starch and starch is carbohydrate? The question is why? Why less than 1? How do you take, or how are these potato chips made? eat those potato chips if they're not fried? And what is that thing in which you fry it? So these potato chips are fried in oil. And what is oil? Oil is fat. So here it is 1 and this is less than 1. So RQ is less than 1. Ayush, it is not 1. Initially you all gave correct answer, less than 1. But when I started asking why, give me the reason, then you shifted to 1. But originally it was correct answer, but whenever you answer, you have to know the reason for it. Alright? Now last one. What is the RQ of milk? If you take a glass of milk without adding sugar into it, nothing is added in that milk, pure milk. Okay, so if you take 1 glass of milk without adding anything into it, no sugar, no Bournvita, nothing, just pure milk, what will be the RQ? Think about it and then give me the answer. 1, less than 1, more than 1. Now everybody is thinking before answering. Less than 1, Ayush has written. What about others? I'm going to ask you why. So be ready with that answer also. Why do you think it is less than 1? Ayush. Milk contains 3.4 grams of proteins. That's why I think it will be less than 1. Is fat also present in milk? Milk is considered as a balanced diet. It has proteins, fats and carbohydrates. So don't you think it is similar to this? It has proteins, it has fats, it has carbohydrates, and that is why RQ is going to be less than 1. Oh, you googled it. That was very fast. And that's why I was wondering how come you have specific information that it has 3.4 grams of proteins. So Google did not give you the amount of fat which is present there. So Google is giving you incomplete information. So rely on your knowledge. Google is going to fool you. It contains proteins, carbohydrates, and that is why milk is considered as a balanced diet. Alright, so now you got an idea how to calculate RQ, whether directly asked or if they give you some kind of a food item. Okay? So whatever we did today, you have understood everything, and see, I'm going to tell you one more important thing about the timing, and I'm going to write it here for you. Tomorrow, Sunday, that is 25th, the timing is 12:30 to 1:30. These changes are going to be only for this the week which is from Sunday onwards. Then 26th to 30th, the timing is going to be 3:30 to 4:30. Okay, and then again on 31st, it is going to be 12:30 to 1:30. All right. After 1st, that is from 1st of November, we won't be changing the timings like this. It is just because I have some important assignments, and that's why. Yes, today's lecture is complete and I hope you have understood everything. Don't forget to take the subscription as early as you can, so that as soon as I start my plus classes, you are able to join them. And when you take the subscription, you can use the code Neela, that is my code, so that you are able to get 10% discount, because from today onwards the prices for the subscription plans have gone up. So I will see you tomorrow, and I have given you the timing, so keep that timing in mind. Tomorrow I'll see you at 12:30. All right, so we will continue with aerobic respiration now. So till tomorrow 12:30, take care, keep revising, okay, and I'll see you tomorrow. Bye.